right, let's talk about substrate supply. So we'll be talking about the process of CO2 getting from atmospheric air into the leaf to the site where it can react with rubisco and ribulose bisphosphate. So one thing I wanted to point out here, and maybe it's a little early, but I'll be talking about, so we talked in the carbon reactions that when rubisco reacts with CO2, that's a carboxylation reaction. And so one thing that's really important in the rate of photosynthesis is the concentration of CO2 at the site of carboxylation. So right where rubisco needs to be grabbing it. So that would be like, what's the concentration of CO2 in the stroma where all of that rubisco is located? And we designate that as C sub C. So that's CO2 concentration, big C, at the site of carboxylation, little c. And we will be using this in some equations in the next couple of weeks. And I might use this term, so I just wanted to highlight it here. All right, so let's talk about the pathway that CO2 has to take to get to the site of carboxylation. So CO2 that's kind of in atmospheric air, it's going to cross over the boundary layer, which we will talk about in just a second, move through the stomate, and then pass through the mesophyll before it then has to enter a cell to get to the actual chloroplast. So I'll just draw this. Well, I don't know if I'll draw it really, but maybe I will. Since I said draw, I'll draw. So boundary layer is going to be a layer of air. We're going to talk about this more in just a second, but it's a layer in air right next to the leaf. And typically this boundary layer, not typically, a boundary layer is a layer of air where the molecules are not moving due to mass flow. Okay, I wasn't going to talk about boundary layer right away, but I guess we'll just jump into it and we'll come back and talk about all of these steps. Okay, so mass flow is when all the molecules are moving. For us, when we're thinking about the atmosphere, we're thinking about wind movement. So wind is moving all of the molecules in the same direction, approximately the same rate. Diffusion is different, right? So diffusion is the movement of molecules from a place of high concentration to low concentration. So ignore the conductances for now. So if we're talking about CO2, all right, this is going to be really bad handwriting because this tablet, I'm not used to it. So let's say this is CO2 in the atmosphere. So I'm just going to use ATM as the atmosphere. So we know that's approximately 400 ppm right now. All right, what's going on? Let me try to get rid of that. Okay, let's try again. So CO2, obviously I'm still working on my drawing skills here. I think I'm having some communication issues with my tablet. All right, try one more time. Maybe we'll give up here. So CO2 in the atmosphere is about 400 ppm. And let's just say I'm going to draw a leaf here with a stomatal opening here. And let's say rubisco is using the CO2 that's diffusing in, so CO2 concentration is 200. All right? So we've got a high concentration of CO2 outside the leaf, low concentration inside the leaf. And so if there's no air movement blowing the molecules around, CO2 is going to diffuse down this concentration gradient and move into the leaf, right? So that's diffusion. That's a relatively slow process. Mass flow is a much faster process, but all of the molecules are moving, in the same direction. Diffusion, molecules can be moving in opposite directions, right? So for example, we've got H2O in relatively high concentration inside the leaf that's leaving and going the opposite direction due to diffusion. All right. So just to highlight that, we'll come back and talk about water more, but just wanted to make sure the difference between mass flow and diffusion were clear. All right, let me go back a slide. Okay, so boundary layer is a layer of air that's where the molecules are not moving very much, where diffusion is the primary process that's making molecules move. Okay, and the thickness of this boundary layer can change a lot. And what we're talking about now is that boundary layer that's right around the leaf surface. So that can change due to lots of different factors. The two we're going to talk about right now are wind speed and the dimension of the leaf. And so this equation that's here is a way we estimate boundary layer conductance. So conductance is the rate that something is moving, right? So a high conductance value means some molecule, in our case, is moving relatively quickly. And a slow conductance means it's moving slowly. So the conductance of a leaf is determined, or estimated, I should say, not determined, estimated by this equation with these two variables, U over D. U stands for wind speed, and D is the dimension of the leaf. And really that's leaf size. Technically speaking, it's the length of the leaf that's parallel to the wind direction. All right, but for us, we can just really think about leaf size. So we're talking big leaves versus little leaves. So let's talk about why those two things are affecting boundary layer conductance. So let's see if I can draw again. Let's say this is our leaf, all right? And I'm going to draw a dotted line that kind of represents the thickness of the boundary layer, all right? Let's pretend this is our boundary layer to start with. If the wind speed is really fast, well, okay, let me back up one step. What creates the boundary layer is the friction of a stationary object acting on something, you know, the air moving across it. And so if the air moving across it is relatively slow, the friction actually has a big impact on that air movement, and it allows a thick boundary layer. But if the wind is moving relatively fast, the force of the friction is relatively small and that boundary layer shrinks. All right, so under high wind speeds, we have a thin boundary layer, and under slow wind speeds, we have a thick boundary layer. And you can see that in this equation. Since wind speed is in the numerator, a large value of the wind speed also makes the product, or boundary layer conductance, a large value. Okay? A thick boundary layer, so to clarify, I think I probably didn't say this clearly, the movement of CO2 across the boundary layer is purely based on diffusion, which, as I said, is kind of a slow process. So if that boundary layer is really thick, you've got a long distance that diffusion has to take place across, and that's going to make it a slow process. So a low value of boundary layer conductance when it's thick. All right? So that's wind speed. I didn't use my drawing at all, but I'll use it now. Okay, let's say we've got a leaf and we've got wind blowing this way across the leaf. And I'm going to draw another leaf that's much smaller. All right? Sorry, those are such horrible leaves. What happens as the wind is blowing across the leaf? Remember, that leaf is relatively stationary, so it's providing, or it's got this resistance or frictional force on the wind movement. And so as air or wind is moving across the leaf, the impact of that friction builds up as the wind moves across the leaf. So what we actually get, remember this dotted line is my boundary layer thickness, is the boundary layer actually gets thicker, you know, the further that the wind is passing across the leaf. I don't know if I can, yeah, all right, sorry. I wanted to draw my boundary layer thickness better. Let me do this. Oops. Went the wrong way. All right. All right, this is our wind. Here's leaf 1. Here's leaf 2. And so as the wind passes across this leaf, we're getting a thicker and thicker boundary layer. So if we were to average the thickness of this boundary layer, it might be something like this thick, right? On this short leaf, it starts out with the same thickness and also builds up. But because it's a much smaller leaf, the average boundary layer thickness is much thinner on this small leaf than on the big leaf. And so that's why the dimension of the leaf is important in estimating boundary layer conductance. Because the boundary layer is relatively thin on a small leaf, a small leaf is going to have faster boundary layer conductance. And the boundary layer conductance of a large leaf is going to be much slower. All right? So boundary layer conductance is really, really important in some systems and some conditions. When you have in the tropics, in the understory in particular, that's one place where we see this, where there's very little air movement because the upper canopy is so dense, we can have these really, really thick boundary layers. And with small leaf plants in really like desert conditions, having that small leaf can be a benefit because it helps cool the leaf and keep it about the same as air temperature versus it heating up. If it has a really, oh sorry, I switched from CO2 to heat. I shouldn't have done that. Scratch the last like 10 seconds of what I just said. And let's just focus on boundary layer conductance as it is. We'll talk more about why boundary layer is so important in the next few weeks. Okay. There's a slide on boundary layer or on conductance and resistance. Let's just ignore it for now, and I'll come back to it. Okay, back to this slide where we're talking about the pathway of CO2. So first, CO2 has to pass through the boundary layer conductance, and we just talked about that. Then it's going to pass through the stomate, and this process of passing through the stomate is still based on diffusion. It's still a diffusional process. The thing that impacts stomatal conductance of a leaf. So if we're not thinking about stomatal conductance of an individual stomate, we're thinking about the whole leaf. And so what affects how fast CO2 is moving through the stomates is how many stomata there are, how big those stomata are, and then how big each individual stomata is, or how wide open it is at a particular time. So I think you can imagine that if you've got lots of really big stomates, that can create a lot of openings in the leaf surface for CO2 to diffuse through. Whereas if you've got just a few small stomates, there's not a lot of openings for CO2 to get through. So that's going to impact how fast it gets into the leaf. I thought I had a drawing, but I don't. And then lastly, the aperture of the stomate is really important. So if you haven't thought about this for a while, okay, this is going to be another horrible drawing. All right, so a stomate is made up of two cells, and these are called guard cells. Again, kindergarten penmanship. All right, and these guard cells can open and close. So this is an example of a stomate that's mostly closed, and then the plant can trigger. They look something like this when they're open. It causes the guard cells to swell, and that swelling actually opens the stomates. And so then when they're open like this is just an airspace, a pore for CO2 to diffuse into the leaf. Of course, we know plants are using water and losing water, and so water is coming out of that pore at the same time when it's open. So how open those stomates are is a function of more than just these three things, but these are the three big things. Light. So in the morning, when light first starts to hit the leaf surface, that triggers the plant to open its stomates, so CO2 can start diffusing in, because now the plant is getting some energy from that light, and it can start doing those carbon reactions. Water availability, as I mentioned, the water's coming out of that stomate at the same time CO2 is getting in. And so plants will close that stomate if it needs to conserve water. And then, of course, that's going to slow down CO2 getting into the leaf. So it's going to reduce stomatal conductance. And then lastly, CO2 supply. If rubisco is, ready to go, it's got plenty of energy, but there's not enough CO2, that can trigger the stomates to open even wider. or as wide as possible to get CO2 into the leaf, into the site of carboxylation faster. And so those are three of the big things controlling the rate of stomatal conductance. All right, let's go back to our figure right here. Excuse me. So boundary layer conductance is that layer of air just outside of the leaf. We just talked about the stomate. So down here is a picture of a stomate with guard cells. For most species, indeed, the stomates are concentrated on the lower surface. Some species only have them on the lower surface. Some have them on both surfaces, but most species have more on the lower surface. And so once CO2 diffuses through the stomate, it still has to get through the mesophyll and through a plant cell wall to get to the chloroplast. Also remember, when we were talking about the light reactions, that this palisade layer up here is where most of the chloroplast are. And we know the carbon reactions are happening inside the chloroplast. So the CO2 has to get from down here, or wherever the stomate is, and diffuse up through the spongy mesophyll to get to that palisade layer. And so that process can be, or that pathway can be relatively long. So depending on how thick the spongy mesophyll is, that can impact the mesophyll conductance. And then once the CO2 gets to a point that it's going to enter a cell, there's several different processes for that molecule to get into the cell. And let's go back to this other slide here on mesophyll conductance. So as I just said, that CO2 has to move from that substomatal cavity of in the leaf, so just inside the stomate, and get to a cell where it's going to enter that cell. It must pass through the cell wall and the cell membranes. And it does that primarily through two mechanisms, through plasmodesmata and aquaporins. Plasmodesmata are just channels in the cell wall. And so some molecules can pass through the plasmodesmata. And so that's one way for CO2 to get into a cell. And then also the aquaporins. So aquaporins are a type of protein that can actually transport small molecules. Aquaporins were named when they were first discovered to transport water. And so that's why they're called aquaporins, but they transport other molecules as well, including CO2. So that's an active transport mechanism. Takes some energy for a plant to move molecules from outside the plant inside the plant. Or sorry, not outside the plant, but outside the cell to inside the cell. And then just due to that concentration gradient, CO2 can move across that cell wall and cell membrane. That's a very, very slow process, but some of the CO2 moves through that way as well. And mesophyll conductance is sensitive. We know it's sensitive to light. We know it's sensitive to CO2 supply. And those two processes are mainly the active transport part of mesophyll conductance. So the part where CO2 is just diffusing through the spongy mesophyll, that's not changing a lot. That's just based on how long that pathway is. But then those aquaporins, they can be upregulated to move molecules faster or slower. And that's what is sensitive to light and CO2 supply. And this area of research looking at mesophyll conductance is like really active right now, and they're discovering lots of new things all the time. And so that's why I put a question mark, is because I think there's other things I'm not familiar with, and there's many other things to discover as far as what's actually controlling that mesophyll conductance. Okay, so let's see. So let's just to review, I guess let's review real quick. Where's my pen? So we've got three main parts of the pathway. We've got boundary layer conductance, stomatal conductance, and mesophyll conductance. And so all three of these things are combining to control the rate that CO2 goes from outside the leaf to where Rubisco is actually reacting with CO2. So it's three separate processes that are all controlled by potentially different external factors, right? And so that the rate that CO2 gets to the point of carboxylation is a really complicated process and is sensitive to lots of different things. And this is where I feel like you could probably argue that the bulk of ecophysiology has been focused on just how fast CO2 gets to the point of carboxylation. There's just tons of research on this, and there's still lots we don't understand. Which, this is what I love and I'm really get excited about, is because we've got these stomates, which are just two cells that open and close, and we spend and have spent decades studying them, and we still don't fully understand them. And so just the complexity of plants and biological life is just so cool. Anyway, side note. Movement of CO2 into the leaf. I don't even know why I have this here, because it's really just a process of these three factors we just talked about. But just understand that full movement and that complexity, and understand what's controlling each individual component of that pathway. And then just making sure you know stomate, what a stomate is, and that stomatal aperture, what some of the things that control that stomatal aperture. We'll continue talking about stomatal conductance and stomatal aperture throughout the semester. And then from an ecophysiological standpoint, interspecific differences in all of these factors. I talked about leaf size being important for boundary layer conductance, so that's certainly an interspecific difference. The number and size of stomates is very different. The thickness of the mesophyll, the number of aquaporins and plasmodesmata, all of those things are different between different species, and so have a huge impact on how fast different species can get CO2 to that site of carboxylation. And then, as I talked about, the sensitivity of those different conductances to the environmental conditions. And so all of these factors are changing over the course of the day and over the course of the season. And so they're really, really dynamic. And I think that's part of the reason we study them so much, is to understand them. What they're doing all the time just involves a lot of research to figure those things out. So we'll spend more time on this, but this is definitely an area where ecophys has been focused for many, many years. Okay, that's all I've got for today. So we're done. Yeah, that's it. We'll see you next week.